Namaste, good morning my dear students. Today let us continue the discussion of cell, the unit of life concept. Under that till the previous cell organelle, the microtubules, then mesosomes, centriole we discussed. Today let us focusing on nucleus and chromosomes. Okay. So, what is meant by nucleus, who discovered the nucleus and the structure, etc. If you take the nucleus concept, nucleus, it was first described by Robert Brown. In 1831. So, Robert Brown is a person who described the nucleus at first in 18th century 1831, and the material of the nucleus stained by the basic dyes was given the name chromatin by Fleming. So, Fleming, person called Fleming, he did the dyeing or coloring or staining of this nucleus. So, nucleus is stained by. So, in the case of our microscopic preparation with respect to slide preparation, he added the basic dye or coloring agent, and that gives color to the nucleus, and he given the name chromatin. Chromatin refers to colored body, colored part of the cell, chromatin, and normally a cell has only one nucleus. So, remember two scientists work. Robert Brown first described the nucleus, and the material of the nucleus will going to get the color or going to capture the color after adding the basic dye, and it was named like chromatin by Fleming because of the coloration observation under microscope. So, we can observe the colored particle, the nucleus, this one in the cell. Then a single cell is having a single nucleus, uninucleate condition. So, usually uninucleate condition. So, one single cell is having one single nucleus. So, cell is with the single nucleus, uninucleate condition, but some of the cells, they may have more than one nucleus. For example, if you take liver cell is having more than one nucleus. Some matured cells lack the nucleus. Some of the cells are having more than one nucleus. So, it is called multinucleate. Some of the cells with no nucleus. So, matured cell. So, in the initial stage the nucleus may be present. When the cell get maturity, the nucleus is not there. So, example for that one is mammalian RBC. Mammalian RBC, and another one is sieve tube cells of vascular plant. Sieve tube cells with respect to our vascular structures of the plant angiosperm, if you take, where the sieve tube cells So, we are studying the sieve tube cells, companion cells, then phloem parenchyma, xylem vessels, xylem tracheus, etc., etc. The vascular tissues are going to be studied in the anatomy of flowering plant in that chapter. Okay. So, in that sieve tube cells of vascular plants, they are not having the nucleus, and mammalian RBC including human RBC, we are having the RBC red blood cells where the matured RBC is not with the nucleus. So, it is enucleated RBC. Human RBC is enucleated, the matured RBC is enucleated. So, nucleus is totally absent. So, that is for the basic concepts of this nucleus. Now, coming to the next point here, if you take the cell cycle refers to the cell division process. So, before the beginning of the cell cycle, there is the interphase, the cell is ready for cell division. So, preparatory phase, preparation for cell division. So, in that interphase, nucleus contain following aspects, because in the mitotic phase or cell division phase, ಅಲ್ಲಿ ಸೆಲ್ ಡಿವಿಷನ್ ಆಗ್ತಾ ಇರುವಾಗ ಇದು ಕೆಲವೆಲ್ಲ ಕಂಡು ಬರೋದಿಲ್ಲ. ಸೊ ಕರೆಕ್ಟ್ ಆದಂತಹ ನ್ಯೂಕ್ಲಿಯಸ್ನ ಪಿಕ್ಚರ್ ಅಂಡರ್ ಮೈಕ್ರೋಸ್ಕೋಪ್ ಅಬ್ಸರ್ವೇಷನ್ ಆಗುವಂಥದ್ದು in the interphase stage, where we can observe nuclear envelope, nuclear matrix, then chromatin and nucleolus. 4 important parts are there. So, nuclear envelope, the covering of the nucleus, nucleus is covered with the 2 nuclear membrane, outer nuclear membrane and inner. So, nuclear envelope, outermost layer covering. Then nuclear matrix, that is the cytoplasm of the nucleus. So, cytoplasm of the nucleus, it is called nucleoplasm. Nucleoplasm, it is the nuclear matrix which is present inner to the nuclear envelope. And chromatin, chromatin refers to the genetic material and the nucleolus. So, these are the basic important nature, that is the parts observed inside the nucleus. So, nuclear envelope, chromatin, nucleolus and the matrix or nucleoplasm. The nuclear pore is present. In the nuclear envelope we can observe the openings, that is called nuclear pore. So, when we are drawing the nucleus, we are making the structure like this. This is one membrane followed with the another membrane, and here you can make out the small openings here and there. This is the nuclear pore, that is important for the movement of materials, or there is a communication between inner part of the nucleus and cytoplasm of the cell. So, for that we can observe a small opening, that is the nuclear pore. So, nuclear pore is present. Nuclear pores are observed on the surface of nuclear envelope. Next, if you take one by one character, nuclear envelope, double layered membrane with a space between 10 to 50 nanometer called perinuclear space. See here, the outer nuclear membrane, next inner nuclear membrane. Between the nuclear outer and inner membrane, we can observe perinuclear space in between outer and inner membrane. ಹೀಗಿದ್ರೆ ಇಲ್ಲಿ ಸ್ಪೇಸ್ ಇರುತ್ತಲ್ವಾ. So, that is what perinuclear space is present. And what about the total width of that space? It is about 10 to 50 nanometer, and it is a barrier between the materials in the nucleus and cytoplasm. Of course, there is a nuclear pore, but the nuclear envelope will act as a barrier, it is a gate between inner nucleoplasm and outer cytoplasm of the cell. ಒಳಗಿನ ನ್ಯೂಕ್ಲಿಯೋಪ್ಲಾಸಮ್, ನ್ಯೂಕ್ಲಿಯರ್ ಮ್ಯಾಟ್ರಿಕ್ಸ್ ಏನಿದೆ ಮತ್ತು ಹೊರಗಿನ ಸೈಟೋಪ್ಲಾಸಮ್ ಏನಿದೆ ಸೆಲ್ಲಿನದು, in between that one it is acting like a barrier between the materials of the nucleus and cytoplasm. And outer membrane usually continuous with the endoplasmic reticulum and bears ribosome on it, that we already studied when we are studying about endoplasmic reticulum. It is the extension of outer nuclear membrane. So, outer nuclear membrane will going to show the extension of endoplasmic reticulum. See, so illinda endoplasmic reticulum ಶುರು ಆಗುತ್ತೆ ಹೀಗೆ like this. So, it is the extension of outer nuclear membrane, and where we can observe the ribosomes. So, protein factories are present. So, it is called rough endoplasmic reticulum. Then nuclear envelope has minute pores formed by the fusion of its two membrane. So, in the side, because of the fusion of inner membrane and outer membrane, we can observe the formation of the pore. That pore formation is there and it is called nuclear pore. So, these are the passages for the movement of RNA and protein between nucleus and cytoplasm. So, movement of RNA, in the beginning I said that transcription, isn't it, where from the DNA which is present in the nucleus is going to give mRNA, and that mRNA will travel through the nuclear pore and reach into the cytoplasm, where the protein synthesis will going to happen, because ribosomes are present in the cytoplasm, they are the protein factories. So, now here the DNA genetic material, or that now it is called chromatin, in the cell cycle cell division phenomena, it will going to form the chromosomes, where genetic material DNA is present, and this chromatin or this genetic material will going to produce mRNA, messenger RNA. That messenger RNA will travel through this nuclear pore and it will come out to the cytoplasm. Cytoplasm barutte. Cytoplasm enirutte, the ribosomes are there. That mRNA is going to be utilized for the production of protein, that is called translation. So, transcription is the production of mRNA from DNA, it is happening inside the nucleus. The newly formed mRNA will come out through that nuclear pore and reach into the cytoplasm, where the site of protein synthesis is there, that is a ribosome. By the utilization of ribosome, the translation phenomena will be there, result in the production of protein. That is the other part of the important part of the molecular biology. So, that the protein synthesis, transcription, translation, etc., that we are studying in the second year topic, okay, second year biology, second PUC biology. Okay. So, here just mentioning of that one, why it is happening, the movement of RNA, which is that major RNA, it is the mRNA, and protein between the nucleus and cytoplasm, the movement is there. So, that is why this nuclear pore. Then that is the nuclear envelope. Second part is the chromatin. What is this chromatin? A network of nucleoprotein fibers. So, chromatin means, what is meant by chromatin? It is the network of nucleoprotein fibers, and it contains DNA and basic protein, that is a histone. So, chromatin is a network. ಅದು ನೆಟ್ವರ್ಕಿನ ಕಾಣುತ್ತೆ, ವೆಬ್ಬಿನ ಕಾಣುತ್ತೆ, ಒಂದು ಬಲೆ ಹಾಗೆ ಕಾಣುತ್ತೆ ಒಟ್ಟು ಅಲ್ವಾ. ಸೊ ಇಟ್ ವಿಲ್ ಗೋಯಿಂಗ್ ಟು ಫಾರ್ಮ್ ಪರ್ಟಿಕ್ಯುಲರ್ ಸ್ಟ್ರಕ್ಚರ್, ದಟ್ ಈಸ್ ವಾಟ್ ನ್ಯೂಕ್ಲಿಯೋಸೋಮ್ ಮಾಡೆಲ್ ಅಂತ ಹೇಳ್ತೀವಿ. ಸೊ ಹಿಯರ್ ಅದು ಏನಿರುತ್ತೆ, ದ ನೆಟ್ವರ್ಕ್ ಆಫ್ ನ್ಯೂಕ್ಲಿಯೋ ಪ್ರೋಟೀನ್ ಫೈಬರ್ಸ್ ಅಂತ ಹೇಳ್ತೀವಿ. ವಾಟ್ ಈಸ್ ದ್ಯಾಟ್? ಇಟ್ ಈಸ್ ಕಂಟೈನಿಂಗ್ DNA. ಸೊ ಕಂಟೈನ್ DNA ಆ್ಯಂಡ್ ಬೇಸಿಕ್ ಪ್ರೋಟೀನ್. ಸೊ DNA ಮತ್ತು ಬೇಸಿಕ್ ಪ್ರೋಟೀನ್ ಇದೆ, ಅದಕ್ಕೆ ನ್ಯೂಕ್ಲಿಯೋ ಪ್ರೋಟೀನ್ ಅಂತ ಹೇಳ್ತೀವಿ. ನ್ಯೂಕ್ಲಿಕ್ ಆಸಿಡ್ DNA, isn't it? Major nucleic acid is the DNA, deoxyribonucleic acid, deoxyribonucleic acid genetic material, and which is associated with the basic protein, that is histone particle, and non-histone proteins and RNA. ಇದೆಲ್ಲವೂ ಕೂಡ ಇದಿಷ್ಟು ಜಾಗದಲ್ಲಿ ಇರುತ್ತೆ. That is what chromatin. Chromatin, network of fibers, so it is called nucleoprotein. So, nucleoproteins are there, and it is with the DNA plus histone protein, and RNA is also there, then non-histone protein, non-histone protein. Okay. So, here the histone protein is present associated with the DNA. Histone proteins are positively charged particle, and DNA is negatively charged particle. So, wrapping of DNA around histone protein is going to be there under chromatin. Okay. That is nucleosome model, we will study that one in the second year. Okay. Now, during cell division, chromatin condenses to form chromosome. What is this condensation? ಕ್ರೊಮ್ಯಾಟಿನ್ ಫೈಬರ್ಸ್ ಏನಿದೆ, ಈಗ ನೋಡಿ ಇಲ್ಲಿ ಉಲನ್ ಬಾಲ್ನಾಗಿದೆ, ಉಲನ್ ಬಾಲ್ ಒಂದು ನೂಲುಂಡೆ ಅಂತ ಹೇಳ್ಬೋದು, ನೂಲಿನದ್ದು ಒಂದು ಬಾಲ್ ಮಾಡಿದರೆ ಹೇಗಾಗ್ಬೋದು, ಆ ರೀತಿ ಉಲನ್ ಬಾಲ್ ಅಂತ ಹೇಳ್ತೀವಿ. ಸೊ ಈ ರೀತಿಯ ಕ್ರೊಮ್ಯಾಟಿನ್ ಫೈಬರ್ಸ್ ಏನಿದೆ ಅದು ಕಂಡೆನ್ಸೇಷನ್ ಆಗುತ್ತೆ. ವಾಟ್ ಈಸ್ ಕಂಡೆನ್ಸೇಷನ್? ಥಿಕ್ನೆಸ್ ವಿಲ್ ಗೋಯಿಂಗ್ ಟು ಇನ್ಕ್ರೀಸ್ ಆ್ಯಂಡ್ ಲೆಂತ್ ವಿಲ್ ಗೋಯಿಂಗ್ ಟು ಡಿಕ್ರೀಸ್. ದಟ್ ಈಸ್ ಕಂಡೆನ್ಸೇಷನ್ ರಿಗಾರ್ಡಿಂಗ್ ವಿತ್ ಕ್ರೊಮೋಸೋಮ್, ರಿಗಾರ್ಡಿಂಗ್ ವಿತ್ ದಿಸ್ chromatin. So chromatin material, that thin fiber like long thread like structure is condenses and going to increase the thickness and forms chromosomes. So, during cell division chromatin condenses to form chromosomes, that is the observation. And second one, sorry, the third one, nucleolus, one or more non-membranous spherical body. So, what is this nucleolus? Non-membranous spherical structure, it continues with the nucleoplasm, it is with the nucleoplasm, this is the nucleolus, this is the chromatin material. So, ಇದೆಲ್ಲವೂ ಕೂಡ ಏನು ಎಲ್ಲಿರುತ್ತೆ ನ್ಯೂಕ್ಲಿಯಸ್ನ ಒಳಗೆ ನ್ಯೂಕ್ಲಿಯೋಪ್ಲಾಸಮ್ ಅಂದರೆ ಸೈಟೋಪ್ಲಾಸಮ್ ಆಫ್ ದ ನ್ಯೂಕ್ಲಿಯಸ್ನಲ್ಲಿ ಇರುತ್ತಲ್ವಾ. ಸೊ ಇಟ್ ಈಸ್ ಕಂಟಿನ್ಯೂಸ್ ವಿತ್ ನ್ಯೂಕ್ಲಿಯೋಪ್ಲಾಸಮ್. ಸೊ ಕ್ರೊಮ್ಯಾಟಿನ್ ಮೆಟೀರಿಯಲ್ ಏನು ಅದು ಒಟ್ಟು ಏನು ಮಾಸಲ್ಲಿರುತ್ತೆ ಥ್ರೆಡ್ ಲೈಕ್ ಸ್ಟ್ರಕ್ಚರ್. ಸೊ ನ್ಯೂಕ್ಲಿಯೋಪ್ಲಾಸಮ್ನಲ್ಲಿ ಇರುವಂತಹ ನಾನ್ ಮೆಂಬ್ರೇನಿಯಸ್ ಸ್ಪೆರಿಕಲ್ ಬಾಡಿ ದಟ್ ಈಸ್ ಕಾಲ್ಡ್ ವಾಟ್ ನ್ಯೂಕ್ಲಿಯೋಲಸ್. It is a site for ribosomal RNA synthesis. It is a site for ribosomal RNA, refers to rRNA. rRNA is the structural component of ribosome. So rRNA synthesis agudu elli, that is the nucleolus. So site for ribosomal RNA synthesis, that is the phenomenon here tagged with our nucleolus. So nucleolus is a non-membranous spherical body present in the nucleoplasm of the nucleus and continuous with the nucleoplasm and going to have the site for ribosomal RNA synthesis. Then the next one, I said in the previous slide that condensation of the chromatin material will going to give chromosomes, isn't it? So condensation of the chromatin in length will going to decrease, thickness will going to increase, result in the formation of that chromosomes. And here a human cell has about 2 meter long DNA. So a chromosomal ಏನಿರುತ್ತೆ DNA ಇರುತ್ತೆ. ಎಷ್ಟು DNA ಇರುತ್ತೆ? What about the amount? It is about 2 meter DNA distributed among 46 chromosomes, means 23 pairs of chromosomes. We all know that in our body we are with 23 pairs of chromosome. So ಎಷ್ಟಿರುತ್ತೆ 23 pair. So 23 pairs ಅಲ್ಲಿ 22 pairs and 1 pair. 22 pairs are autosomes and 1 pair it is the sex chromosome. 1 pair is the sex chromosome that will determine the sex of an individual. So in the case of female it is sex chromosome. In that female we can have the representation XX. In the case of male the representation as XY, that is the representation of chromosome with respect to male and female. So that is what 46 chromosome or 23 pairs of chromosome. Then every chromosome has a primary constriction. We can observe one constricted area that is called centromeric or centromere region, centromeric region. So constricted area, it will going to form the constricted area on the side of which disc shaped structure that is present, it is called a kinetochore. Here are given, yeah, kinetochore. In the side of that constricted area of chromosome, it is with the kinetochore. So kinetochores are present and this centromere with the kinetochore, that is the nature of this chromosomes. Then the other part, ಇದು ಈಗ ನಾವು ಡಯಾಗ್ರಾಮೆಟಿಕ್, ಇದು ಇನ್ನೊಂದು ವ್ಯೂ ಅಷ್ಟೇ ಕೈನೆಟೋಕೋರ್ನದು. And other part mitotic spindle formation, that is the part of cell cycle. Then sister chromatid, the arm of the chromosome, it is called sister chromatid, it is called chromatids. So it is identical, so it is called sister chromatid with respect to that chromosome. Then based on the position of centromere, central part, constricted part of the chromosome, that is the centromere, chromosomes are of 4 types. 4 ಟೈಪ್ ಅಲ್ಲಿ chromosomes ಅನ್ನು ಕ್ಲಾಸಿಫಿಕೇಶನ್ ಮಾಡಬಹುದು. 4 types of classification with respect to chromosome based on the position of centromere in the chromosome. One is called metacentric chromosome, middle centromere forming 2 equal arm of the chromosome. This is the metacentric. Middle alli centromere irutte with the equal arms. Then it is called metacentric chromosome. Then submetacentric, second one, submetacentric. What is this submetacentric? Here centromere is present but nearer to one end forming one short arm, another one is long arm. Almost near to the middle but going to have one short arm and one set of long arm. Then it is called submetacentric chromosome. This is the submetacentric chromosome. It is exact nodi, this length and this length is equal. That is metacentric, middle, meta means middle here, and the centromere is in the middle, exact middle of the entire chromosome. Here we can observe one shorter arm and this is the longer arm. Of course they will going to form secondary construction etc. ಸ್ವಲ್ಪ detail ಆಗಿದೆ, ಅದಷ್ಟು ನಮಗೆ ಬೇಡ. Okay, so one shorter arm and one longer arm. And third one is the acrocentric. Acrocentric, here one very short arm and another one is very long arm. Another set is very long arm. Centromere is close to its end forming one very short arm and one very long arm. So centromere, illi nodi, centromere close to one end with a very short arm set and another one is very long arm set. And telocentric chromosome, terminal centromere. This is the telocentric. Centromere is terminal and here long arm short arm antilla. So only a single set of arm with the terminal centromere, then it is called telocentric chromosome. So remember, this is one of the important question for your exam with the CET or NEET or board exam etc. Metacentric, submetacentric, acrocentric and telocentric chromosome, okay, based on position of centromere in that chromosome. So some chromosomes have non-staining secondary construction at a constant location, it is called satellite. Non-staining, ಈಗ ನೋಡಿ ಕ್ರೋಮೋಸೋಮ್ಸ್ ಏನು ಅಲ್ಲಿ ನಮ್ಮ ಫ್ಲೆಮಿಂಗ್ ಹೇಳಿದ್ದಾನೆ ಏನು ಕ್ರೊಮ್ಯಾಟಿನ್ ಅಂತ, ಅದು ಕಲರನ್ನು ತಗೊಳ್ತದೆ ಅಲ್ವಾ. ಸೊ ಇಟ್ ವಿಲ್ ಗೋಯಿಂಗ್ ಟು ಟೇಕ್ ದ ಕಲರ್ ವಿಚ್ ಇಸ್ ಗೋಯಿಂಗ್ ಟು ಬಿ ದೇರ್ ಇನ್ಸೈಡ್ ದ ನ್ಯೂಕ್ಲಿಯಸ್. ಸೊ ನ್ಯೂಕ್ಲಿಯಸ್ ವಿಲ್ ಗೋಯಿಂಗ್ ಟು ಗೆಟ್ ದ ಕಲರ್, ದಟ್ ಬೇಸಿಕ್ ಡೈ. So some part of the chromosome is not going to take the color, it is called in a particular location, it is called satellite particle. So here we can make out that satellite here. After secondary construction we can observe the satellite particle. So some areas, not in all area, and in the chromosome particular site is there, it is called loci. It is called loci, where the genetic material or the DNA is located. Chromosome entire part of DNA irala, some part is going to have the DNA, that is the location of the chromosome where the DNA is there, where genetic material is there, it is called loci. So that is what another important character. See, this is another picture ಅಷ್ಟೇ, ಏನು ತಲೆಬಿಸಿ ಮಾಡಬೇಡಿ, ಭಯಂಕರ ದೊಡ್ಡ ಉಂಟು ಅಂತ. ಪ್ರೀವಿಯಸ್ ಪಿಕ್ಚರ್ ಅಂತ ಇನ್ನೊಂದು ರೀತಿಯಲ್ಲಿ ಕೊಟ್ಟದಷ್ಟೇ. ಅದು ಪಿ ಆರ್ಮ್ ಕ್ಯೂ ಆರ್ಮ್ ಅಲ್ಲಿದೆ, ಶಾರ್ಟ್ ಆರ್ಮ್ ಲಾಂಗ್ ಆರ್ಮ್, ಡೋಂಟ್ ವರಿ, ಯು ಕ್ಯಾನ್ ರೈಟ್ ಲಾಂಗ್ ಆರ್ಮ್ ಆ್ಯಂಡ್ ಶಾರ್ಟ್ ಆರ್ಮ್, ದಟ್ಸ್ ಇನಫ್. ಓಕೆ, ಸೊ ಮೆಟಾಸೆಂಟ್ರಿಕ್ ಇದು ಆ್ಯಕ್ಚುಲಿ both arms are equal. Submetacentric, one short arm, one longer arm. Acrocentric, one very short arm and one very long arm. Telocentric, terminal centromere. This is the 4 different types of chromosome. See, this is another view of that one. centromere, position, submetacentric, metacentric, acrocentric and telocentric chromosomes. Okay. ಈಗ ನೋಡಿ ಮೈಗ್ರೇಷನ್ ಆಗುವಾಗ ಡ್ಯೂರಿಂಗ್ ದ ಟೈಮ್ ಆಫ್ ಎನಾಫೇಸ್ ಸ್ಟೇಜ್ ಅಂದರೆ ಮೈಟಾಸಿಸ್ನಲ್ಲಿ ಎನಾಫೇಸ್ ಸ್ಟೇಜ್ ಅಂತ ಬರುತ್ತೆ, ಆಗ ಕ್ರೊಮ್ಯಾಟಿಡ್ಸ್ ಸಪರೇಟ್ ಆಗುತ್ತೆ. ಸೊ ಹೌ ಇಟ್ ಲುಕ್ಸ್ with respect to metacentric, with respect to that is the submetacentric, and this is the acrocentric and this is the telocentric. chromosomes na chromatids arm of the chromosome ಹೇಗೆ ಸಪರೇಟ್ ಆಗುತ್ತೆ ಅದನ್ನು ಕೊಟ್ಟದ್ದು, ಅದು ನಮ್ಮ ಸೆಲ್ ಸೈಕಲ್ ಆ್ಯಂಡ್ ಸೆಲ್ ಡಿವಿಷನ್ ಚಾಪ್ಟರ್ಲಿ ಬರುತ್ತೆ ಮಕ್ಕಳು, ಆಯಿತಾ. The next one is the microbodies. These are the membrane bound minute vesicles that contain various enzymes. What is meant by microbodies? These are the membrane bound structures, very minute, very small, very pin like very minute structure, that is a vesicular structure containing various enzymes. Different types of enzymes are present. So crystalline core is present with the lipid bilayer, that is the plasma membrane. Inside that one we can observe the enzymes. Present in both plant and animal cell, because it is the area where the chromosomes are the enzymes are present. Then comparison or difference between plant and animal cell. Plant cell wall present, already discussed, but now once again recalling us. Cell wall is present, made up of cellulose, major content. Animal cell, their cell wall is totally absent. Plastids are present. Plastids ಅಂದ್ರೆ ಏನು? ಕ್ಲೋರೋಪ್ಲಾಸ್ಟ್, ಕ್ರೋಮೋಪ್ಲಾಸ್ಟ್, ಅಲ್ಲೋರೋಪ್ಲಾಸ್ಟ್ ಎಲ್ಲ ಹೇಳಿದ್ದೇನಲ್ಲ ಅದು ಪ್ಲಾಸ್ಟಿಡ್ಸ್. ಅಂದರೆ ನಾವು ಮೈನ್ ಆಗಿ ನಾವು ಫೋಟೋಸಿಂಥಸಿಸ್ಗೆ ಫೋಕಸ್ ಮಾಡುವ. ಪ್ಲಾಸ್ಟಿಡ್ಸ್ ಆರ್ ಪ್ರೆಸೆಂಟ್ ಹಿಯರ್. ಎನಿಮಲ್ ಸೆಲ್ plastids are totally absent. Then a large central vacuole is present in the plant cell. Here large single vacuole is present, that is the plant cell. Here vacuoles are there, but they are many small vacuoles. Small vacuoles are scattered in the entire area. Then centrioles are absent. In the plant cell centrioles are absent. Here centrioles are present in the animal cell. That is the major differences between plant cell and animal cell. Make a note of it, one of the important question. Okay. Then prokaryotic and eukaryotic cell differences, another important question. Prokaryotic cell generally smaller in size, bacteria etc. But eukaryotic cell generally larger or bigger in size. Genetic material is in the form of nucleoid. In the case of prokaryotic cell, clear nucleus is absent because the genetic material is not covered with the nuclear membrane, it is the nucleoid. And in the eukaryotic cell, in the form of nucleolus, in the form of nucleus, the genetic material is present in a clear manner, and the nucleus is with the nuclear membrane. The nuclear membrane is absent in the prokaryote, here it is present. So eukaryotically nucleoid anta helike illa, nucleus anta helodu, prokaryotically nucleoid anta mention martivi. Then membrane bound organelles are absent in the case of prokaryotic cell. Membrane bound organelles are present, like Golgi bodies, endoplasmic reticulum, the plastids, the chloroplast, mitochondria etc. etc. Then circular DNA is present in the case of prokaryotic cell. In the eukaryotic cell, linear DNA is present. DNA is linear chain of nucleotide bases. Then ribosomes are of 70S type. 70S type divided into 50S and 30S, isn't it? Whereas in the case of eukaryotic cell, ribosome is about 80S type. It is divided into 60S and 40S subunits, and the 70S type is there in the case of plastids. So chloroplast and mitochondria, they are having their own ribosome, their own DNA, isn't it? So mitochondria ಮತ್ತು plastid cell, plastids here chloroplast example ತಗೊಳ್ಳುವ, easy to understand. So in the case of plastids and in the case of mitochondria, we can observe 70S type of ribosome, but in that cell, in the eukaryotic cell, in the cytoplasm, it is 80S type of ribosomes. So these are the major differences between plant cell and animal cell. So my dear friends, up to here it is a cell, the unit of life. So we end we came to the end of this chapter, and if you have any doubt in the live discussion, we will going to talk. Okay. So, in the live discussion of this chapter, during that time you can ask if you have any doubt. So, this is what the cell, the unit of life chapter. Okay. If you have any doubt, you can ask through WhatsApp or in the live discussion. Okay. We will provide the notes. Okay. We will provide the notes through WhatsApp, and if you have any doubt, you can ask in the live discussion. So, thank you my dear students.